Alrighty, membrane fans, here we are. We're going to talk about the fluid mosaic model of the phospholipid membrane. Now, come on, it's not that bad. We've already talked a little bit about this, right? The lipid bilayer with lots of proteins in it, right? So it's not just lipids. We talked about phospholipids, we talked about cholesterol. Yes, those are all lipids, but we also have lots and lots of proteins within our phospholipid membrane for our plasma membranes and all of our membranes and all of our organelles. Remember, not just the plasma membrane, but all the organelles have a fluid mosaic membrane. So here we go. Here's a little picture, right? If we are talking about outside of the cell, out here, right, this would be the extracellular, this would be intracellular or the cytoplasm down here, phospholipid membrane with lots of membrane proteins in between. Notice how the membrane proteins pass through the entire thing. Some parts stick out, some parts stick in, right? Some of them are only sticking out one side or just sticking in one side. Amphipathic, that means crossing the membrane. Hydrophobic, hydrophilic. Has to be in order for it to sit like that. Integral proteins mean they're inside, right? Within the membrane. They're amphipathic, span the membrane, both interior and exterior surfaces. We can also call them transmembrane proteins because they trans across the membrane. Okay, so a transmembrane protein, by definition, is an integral protein, right? Sometimes it can help be a transport protein, help stuff across the membrane that can't get across by itself, like the big stuff, sugars that we talked about, can affect membrane permeability. So integral proteins are by definition amphipathic, they span the membrane. What about peripheral proteins? Those guys are ones that are found on only one side of the membrane. Sometimes they're attached to an integral membrane protein and sometimes they're not, but they only face one way. Amphipathic proteins, so what would this kind be? Would this be peripheral or would it be integral? Usually integral. Okay. Oops, there we go. And what does that mean specifically, right? We have some amino acids that are polar or charged, hydrophilic. You can go look those guys up. Hydrophilic R groups, hydrophobic R groups, hydrophilic R groups. This stuff has to hide within the tails. This has to stick out by the heads of the phospholipids. By definition, by chemistry, it has no option. So here we go. Here's it drawn just like we showed. Hydrophilic, outside the cell, inside the cell, hydrophobic, hiding here within this very hydrophobic region. Right? What are these guys? They're a little crooked, so they have to be unsaturated. Fatty acid tails. So we call it the fluid mosaic because things move around. They're not just stuck there. A dynamic mosaic of phospholipids and proteins, right? They're not stuck. This guy can travel through the membrane. This guy can travel through the membrane. This part can't dip down into here, but this whole fat guy can move across closer to this one, closer to this one. They can move. The lipids are free to diffuse rapidly throughout the plane, but they rarely flip. Okay, they rarely, this guy flip down here, this guy flip up here. But they can travel around as long as the hydrophobic regions are here and the hydrophilic regions are there. So there you have it. That's the fluid mosaic model. We get proteins that move around through here, phospholipids that can travel around, usually not flipping places between the layers. Right, and this guy usually not flipping upside down, right? He's stuck here. His hydrophobic is stuck in the hydrophobic, hydrophilic. Here's a peripheral, right? He might be stuck to one of the integrals, but he's just on top. So the peripheral membrane protein, in this case, what would it be? The whole thing would be hydrophilic, right? It's only up there, here, in the area of hydrophilicity, right? At the heads, in the outside. Here's another guy who's peripheral. This guy looks peripheral. This one, amphipathic. Hydrophobic, hydrophilic, hydrophobic, hydrophilic. And there you have it. That's the fluid mosaic model of the membrane structure. Woohoo! So that's the fluid mosaic model.